Welcome back to the Cramming for Biology series. This is video 3 out of 9 for part 2 of bio class. Bear with me if I just don't seem the slightest bit excited. That's right, today we talk about photosynthesis. I apologize in advance for how bare bones this video is about to be, but I absolutely cannot salvage enough motivation within me to want to talk about photosynthesis. Alright, so let's just jump into it. Within photosynthesis, we have the light reaction and the dark reaction. The light reaction is what I will detail first. Okay, the light reaction is where the plant produces energy in the form of ATP, and plants do this through an organelle called a chloroplast. Okay, we don't have chloroplasts as animals, just plants. So within the chloroplast, we have pigments, chlorophylls and carotenoids, that function as photon receptors. Now, photon is just the particle of light energy. And these pigments are concentrated within light harvesting complexes. And the first of those complexes in photosynthesis is light harvesting complex 2. Here, 12 chlorophylls and 2 carotenoids harvest light and pass that photon light energy from light complex to light complex until it reaches the reaction center, which concentrates the light energy and excites electrons. After light complex 2, the excited electrons rush to photosystem 2, where they are excited by pigment P680 to an even higher energy state. Once they're excited, the electrons go through a series of carriers, and they lose energy as they do this, which allows for the pumping of hydrogen ions from the chloroplast stroma to the thylakoid lumen. Now, doesn't that sound kind of familiar? Now, the electrons have lost some energy, and so they have to be re-energized. They are re-energized at photosystem 1's reaction center by the pigment P700, and they are re-energized by photon light energy. Now, why is photosystem 2 before photosystem 1? I don't care. Science is dumb sometimes. After photosystem 1, the electrons once again go through energy carriers, lose energy, and more hydrogens are pumped into the lumen. And then NADP plus finally accepts the electrons and turns into NADPH. Notice how there is now a proton gradient in the thylakoid lumen. Doesn't that sound familiar? That proton gradient, just like we saw in the electron transport chain, is used to drive ATP synthesis in the plant cell through the chloroplast ATP synthase. That is the light reaction. Now, I don't think the light reaction is too bad. I actually think that's kind of cool. But I find the dark reaction to be the biggest thorn in my foot. I'm going to preface this section of the video with the fact that this is just a crap ton of steps for you to memorize. So if you love plants and photosynthesis, props to you. Here's where I mentally check out. The dark reaction, also called the Calvin. Guys, I'm editing this video right now. I am not qualified enough to give you guys the Calvin cycle lecture because I don't understand it myself. I apologize sincerely, but you're on your own for this one. We're just gonna skip right to Rubisco and why it sucks. It is widely known that Rubisco sucks. Though it is an important gateway for carbon to really enter into the ecosystem, it is so slow and inefficient. Funny enough though, it actually makes up for this inefficiency for being crazy abundant. It's actually the most abundant protein in the world. So, like, go Rubisco, I guess. Aside from photosynthesis, you should also understand the difference between C3, C4, and CAM plants. I cannot go into detail here because I myself only have a very baseline knowledge about this, but in C3 plants, the first intermediate in carbon fixation is 3-PGA. The C4 plants split carbon fixation into 2 steps, and CAM plants separate the light and dark reactions into different times of the day to lose as little water as possible. I pinky promise the rest of my lecture videos are going to have way more effort put into them. Photosynthesis, especially the dark reaction, is just not my thing. My understanding of the Calvin cycle is not really there. My bad. With that being said, we have finished today's content for this video, and here's your routine disclaimer. Number 1, I'm a student, not a professor. Number 2, my biology knowledge is by no means perfect. And number 3, I'm making this series using my college notes. If you find an error, please, please, please fact check and correct me. Alright, I love you guys. Thank you so much for putting up with this, and I will see you in the next video where we talk about the nucleus.